what's the deal with cryonics or cryopreservation, and what's the relationship and differences between these two words? Can you explain it to our audiences?
Sure. Well, cryopreservation is a very broad term about preservation of biological materials at low temperatures. That's the concept of cryopreservation. It can be applied to cells, it can be applied to tissues, it can be applied to small organs so far. So it's typically small tissues and cells that can be cryopreserved. So that's broadly speaking, that's cryopreservation. The idea behind cryonics is declared legally dead, they're also cryopreserved. No, while cryonics is about applying cryopreservation to a whole human being, which may or may not work in the future.
And what are the practical applications of cryobiology that are already being used today?
So there's a lot of applications. So this inability of cells to replicate after about 50 population doublings. Eventually they become senescent. And this was discovered in a type of embryonic cell, embryonic fibroblasts called WI38 by a man called Leonard Hayflick, who cryopreserved these cells in the 60s, so over 60 years ago. And the cells that he studied in the 60s, they're cryopreserved, and you can thaw them and you can still use them. So that's one application in biomedical research. We have cells that we can cryopreserve and then come back to them and study them in the future, share them with other labs and so on. So it's very widely used for research, cryopreservation. It's also used in reproductive medicine, IVF, for example. There are people alive now that were frozen as embryos for years in some cases, and then they were thawed and then they were implanted, and now they're perfectly normal individuals, even though they were actually cryopreserved for sometimes years during their early life. So there's a lot of applications now, I suppose, in terms of regenerative medicine as well. There's also application in terms of cryopreserving small tissues, cryopreserving biomaterials, cryopreserving stem cells. So there's a lot of applications in terms of small tissues and cells already. The problem, I should say, is that it's a problem of size. So once you can cryopreserve cells and you can cryopreserve small tissues, but larger tissues take longer. The temperature just takes longer to drop, and so you cannot safely cryopreserve a human liver or a human heart or any large human organ. You cannot cryopreserve it. So and that's a big problem.
Is it a major challenge that we face in the cryopreservation technology now?
Absolutely, because one of the problems we have in medicine is lack of organs for transplants. There's lots of people that need organs for transplants. The problem is that when there is when someone dies in an accident, for example, and they're an organ donor, those organs cannot be retained in some cases. They can have a viability for 24 hours or 48 hours, and so you have that amount of time to find a suitable donor and then ship the organs across the country to that person. And so there's a lot of wastage of organs because of that inability to cryopreserve human organs. So if we could safely cryopreserve organs, like hearts, liver, lungs, kidneys, and so on, if we could safely cryopreserve them, this would save thousands of lives each year, because it would make the whole process much more efficient, and would result in these patients that are dying because they don't have a suitable organ, that they would be able to be rescued.
Then, according to solve this problem, what kind of efforts that we have paid, or do we have any solutions?
So we do know, so it is possible to cryopreserve, as I say, small tissues and cells. So to do this, so one problem that happens is that if you lower the temperature below zero, water freezes, right? And we're mostly water. So one big problem is that once water freezes, it creates crystals. You have ice crystals, and ice crystals are very, I mean, they're sharp essentially, and they will damage the cells and the organs. So, I mean, decades ago, scientists discover cryoprotectants or antifreeze, basically. So these are essentially substances like glycerol and ethylene glycol and DMSO that prevent ice formation. So when you cryopreserve cells, for example, that's what you do. You replace some of the water with an antifreeze, and same for small tissues and structures. So you use these cryoprotectants. The problem is that the cryoprotectants also have some toxicity. So they're low toxic. That's why you use them, but they, at certain concentrations, they start to become more toxic. So, we have some methods to cryopreserve biological structures, but they're still limited. And so what we and others are trying to do is try to overcome that, develop new cryoprotectants, develop new formulations that minimize toxicity, and so on. So there's a lot of efforts in that area in terms of better cryopreserving organs. Ultimately, that's the goal.
We cannot figure out what is the differences between vitrification itself and the traditional freezing technology. And could you please explain it to our audiences about that?
Sure. So there's different methods for cryopreservation, as you mentioned. I mean, there's something called flash freezing, which can be used for cells, where you essentially take cells and you put them in liquid nitrogen. So, ultra low temperatures, and they freeze very rapidly. So that's one method. So you have methods of very fast freezing. And then you have vitrification. Vitrification is a particular method where you have very high concentrations of cryoprotectants. And so because you have very high concentration, you have this viscous solution that never really becomes solid. It becomes like this glass-like structure. But the advantage is that it minimizes ice formation. As I said, ice, is very sharp and can damage. So with vitrification, you don't have ice. Well, if it's done properly, you don't have ice formation. And so vitrification is essentially a process of slow freezing, but with very high concentrations of cryoprotectants, and then that you get this this process where it's not really freezing. It's called this glass-like structure. So vitrification actually means turn to ice. So in vitrification, that's the preferred process. In cryonics, the companies and institutions that do cryonics, they use vitrification.